Hey everyone, welcome back to Neurobiology at Providence College. I'm Joe DeGeorgis. Today we're going to look at a paper by Stanley Prusiner, and this is a continuation of the work by Gajuzek, who thought that Kuru was caused by a slow-acting virus. Prusiner was interested in trying to figure out what the infectious particle was, whether it was indeed a virus or some other type of molecule, and his work was criticized by the scientific community for many years because scientists bought into the slow-acting virus theory and didn't want to believe what Prusiner eventually proved, which is Kuru and other related diseases are actually caused by a protein that he named prion. But eventually Stanley Prusiner proved that indeed it was a protein. That's what this paper argues. And eventually he was awarded the Nobel Prize for this work. So it's very interesting, and I had an opportunity to hear him speak at the Marine Biological Laboratory as part of their Friday evening lecture series, which occurs in the summertime. So I saw him about 6 or 7 years ago. As we already know, based on Gajuzek's work, Kuru could be transmitted from human to chimpanzee and from chimpanzee to chimpanzee by taking diseased brain tissue and grinding it up and injecting it into the brain of the chimp. And it was thought that Kuru was transmitted from human to human through cannibalistic rituals. In this paper, Prusiner starts out by talking about another form of a spongiform disorder similar to Kuru that occurs in sheep and goat called scrapie. So in the first part of the paper, Stanley Prusiner tells a story about inoculating sheep in Scotland against looping ill virus. And what the farmers did is they took brain and spleen tissue from a sheep that died of looping ill virus. And of course those tissues contain the virus. They grind the tissue up and then they treat the tissue with formalin, which kills the virus, and then they inject that slurry into sheep so that the sheep's immune system will recognize this now dead form of the virus and build antibodies against the virus, and of course create immunity within the sheep population. But it turns out that in the process of inoculating the sheep against looping ill, they also injected a second substance unknowingly that was also in the brain or spleen or both from that original sheep, and that caused scrapie in the inoculated sheep population. So okay, along with Kuru, there are other forms of what are now known as prion diseases in humans. One is called Creutzfeldt-Jacobs disease, and of course we already know about fatal familial insomnia, which we're going to get to in tomorrow's lecture. There's scrapie in sheep and goats, there's mad cow disease in cows, and so on. But these are all the exact same disease, just in different organisms. It looks as though the disease can be transmitted through ingestion, that is cannibalism in the 4 tribe. Also, mad cow disease is known to have been caused by cows eating diseased cow material. So it turns out that farmers take parts of the cow that can't be sold and they grind it back up into the feed of the cows. And that's how mad cow disease was transmitted. We know that these diseases can be transmitted by injecting disease material into brain, human to chimp or chimp to chimp. We know that the disease can be transmitted by injecting disease material into the body cavity in the case of scrapie. And there are examples where Creutzfeldt-Jacobs disease has been transmitted from one patient to another patient through the surgical instrumentation. That is to say, a brain surgery was done on a Creutzfeldt-Jacobs individual, and then that instrumentation, even though it's cleaned and sterilized, is used subsequently on other patients. And in the process, the disease was transmitted from the original patient to the downstream patients. So it's a pretty scary thought. At any rate, what all of this shows is that whatever the infectious agent is, that it's quite resilient. And Prusiner set out to figure out what that infectious agent actually is. The paper by Prusiner is a summary of a lot of work, decades of work by the scientific community in this general area. And in order to try to figure out what the infectious particle is, you have to try to biochemically purify it, which is what Prusiner wants to do. The difficulty is that you have to have some sort of assay so that you can follow the particle through the purification procedure. So let me erase this. So, if we were talking about kinesin, we can follow kinesin's purification through either the microtubule gliding assay, or you could do it by binding the material that you're purifying, the fraction of the purification material, bind it to glass beads and see if you can get the glass beads to move along the microtubule. So, for instance, what I mean to say is that if you took a sample and you put it over a sucrose step gradient, like a 45, 15, 12, and then a supernatant, and you put the material on top of the sucrose and then spin it through high speed, of course we know that different materials are going to separate out into the different layers based on their density. It's a sucrose density gradient. And then you would literally have to test each fraction for kinesin in that example, in the kinesin example. And if you found kinesin in, say, the 45% layer, then you put a slide down in some microtubules, and if they glide around on the surface of the glass, or a glass bead incubated with the material moved along the microtubule, then you know kinesin's in this particular fraction. And if it's not, if the movement doesn't happen in the presence of these three other fractions, you could throw those away, and then you would continue your purification from the sample that contains the kinesin. You might then take this fraction, and put it over a sizing column. We've talked about this before, that might have beads with holes in them that separate things based on their size. This was based on density, and now we're separating the sample, the components of the sample based on size. And we said that the small stuff goes into the bead and gets caught there for a little while and then comes out. The big stuff just can't make it in, goes around, and the medium stuff goes into some bigger holes and comes out a little faster than the small stuff that gets stuck in the nooks and crannies of the bead. At any rate, then if we collected fractions as the sample comes off of the column, you would have to test then each of these fractions for kinesin in this particular example. But still, not a very difficult assay. You just add the fraction to, let's say, the glass beads, and if there's kinesin present in the fraction, it binds to the glass bead. And then if you add the beads coated with kinesin to the microtubule in the presence of ATP, you'll see the glass bead move. And if it doesn't move, then that particular fraction didn't contain kinesin. And so you would take that fraction, or those few fractions that do contain the kinesin, and use those to continue with the purification until in the end you have totally purified, squeaky clean kinesin. And you can determine that by running it on a protein gel and seeing that there's only one protein there in the fraction. In the case of trying to purify the scrapie agent, the scientists needed an assay synonymous with the microtubule gliding or the bead movement assay, so that they could determine where the infectious agent was in different fractions that they generated through their biochemistry. They knew from Gajaszek's experiments that it took 1.5 years for chimpanzees to show symptoms of Kuru when the chimpanzees' brains were injected with Kuru brain from human. And that time period was reduced, you'll recall, to 1 year when they injected diseased brain tissue from chimp to chimp. But 1 year is a long time for a bioassay. It turns out that Prusiner was able to develop a hamster model, and these hamsters developed symptoms of the disease 60 to 70 days after injection. But still, you need to take every single fraction that you generate in the biochemistry, and you have to inject it, each one of those, into its own hamster brain, and then you have to wait the 70 days to see which fraction contains the infectious agent. So there's a lot of animals, a lot of time, and a lot of work to pull off this experiment. The second problem is that it turns out when they try to biochemically purify the substance, so let me redraw a gradient. The gradients I've been drawing and we've been talking about so far in the class are called step gradients because we have a 45% and it's a layer, and then on top of that we put a 15% sucrose layer, and you just pipette the layers one on top of each other. It's pretty simple, but it turns out you can also generate a gradient that doesn't have steps in it, but just gradually diminishes in concentration. So you could have a 45% down here, and then say up here at the top it could be 5%, and then right below it is 6%, 7%, and 8%, and 9%. It's just continually increases in concentration. There's sort of a complicated way of it's a machine that generates this. You put a tube in here and you float the 5% first, comes through the tube, and then it gradually increases and the 5% moves up until the tube is complete and you have 5% here and, like I said, 45% down here. So it's a continuous sucrose gradient. At any rate, they used a variety of different techniques to try to biochemically purify the scrapie agent. And like I said, at the end of the day, you have to inject each fraction into the brain of the hamster, and then you have to wait the 70 days to see whether the hamster comes down with the symptoms or not. So you'd have to place the sample onto the gradient and then spin the gradient in a centrifuge, and then the different fractions would come into different spots along the continuous gradient, and then you can puncture a hole in the bottom of the test tube and collect fractions. So the first fraction would be the most concentrated stuff, near 45%, and then the next tube would be a little less concentrated, 44%, and so on. But at the end of the day, each one of these fractions has to be injected into a hamster, and you have to wait the 70-day period to figure out which sample contains the transmissible agent, that is, which one causes scrapie in the hamster. The problem is that they found that the scrapie agent is all over the place. It's in every fraction. And we'll see that there's an explanation for that in tomorrow's paper where we talk about fatal familial insomnia. I'll give you the upshot now. What happens is that the prion, which we now know is a protein, prion protein, they bind to one another and form aggregates. And if you have just a few molecules that are coming together to form an aggregate, like 4 or 5 molecules, then they're pretty small. And if they're made up of 50 or 100 individual proteins making up the aggregate, then those are heavier, and then you can have some in the middle. So you end up with this protein that's really hard to purify because it aggregates with itself, with other members of the same protein or gene product, and it forms different size aggregates in the sample. We know that now. That wasn't known at the time that Prusiner wrote this article. So at any rate, there was a problem with the biochemistry that they couldn't easily purify it. But they did find a sample that was enriched for it. There was more of the transmissible agent in that fraction than in some of the others. And they removed that sample, and they used that sample to try to assay what the nature of the transmissible agent is. So if we have our vial that contains the transmissible agent at high concentration, they say that there are 6 separate lines of evidence that suggest that the transmissible agent contains a protein. One is proteinase K. So this is a protease which cleaves the protein up into small amino acid sequences. So you're literally chopping it up. You can modify the protein with diethyl pyrocarbonate. You can denature it with SDS, which is a detergent. Inactivate it with chaotropic salts. This is like potassium iodide. It breaks up the proteins, breaks up the structure. Inactivate it with phenol, or inactivate it with urea. All of these take apart secondary and tertiary structures of proteins. So if you treat this sample with any of these 6 chemicals that alter protein structure, and then you inject each of those samples into its own hamster, proteinase K, diethyl pyrocarbonate, SDS, and so on, none of these hamsters get scrapey. So by destroying the protein in the sample, you destroy the transmissible agent. Then they go on to treat the sample with RNases, things that destroy RNAs or DNAs, DNase or an RNase, and some other reagents that attack these 2 types of molecules. And when they inject those into the hamsters, they still get the disease. So it looks like the transmissible agent does not contain RNAs or DNAs. It contains protein. So if we have our vial that contains the transmissible agent, they say that there are 6 6 So if we have our vial that contains the transmissible agent at high concentration, they say that there are 6 separate lines of evidence that suggest that the transmissible agent contains a protein. One is proteinase K. So this is a protease which cleaves the protein up into small amino acid sequences. So you're literally chopping it up. You can modify the protein with diethyl pyrocarbonate. You can denature it with SDS, which is a detergent. Inactivate it with chaotropic salts. This is like potassium iodide. It breaks up the proteins, breaks up the structure. Inactivate it with phenol, or inactivate it with urea. All of these take apart secondary structures. All of these take apart secondary and tertiary structures of proteins. So if you treat this sample with any of these 6 chemicals that alter protein structure, and then you inject each of those samples into its own hamster, proteinase K, diethyl pyrocarbonate, SDS, and so on, none of these hamsters get scrapey. So by destroying the protein in the sample, you destroy the transmissible agent. Then they go on to treat the sample with RNases, things that destroy RNAs or DNase, DNase or an RNase, and some other reagents that attack these 2 types of molecules. And when they inject those into the hamsters, they still get the disease. So it looks like the transmissible agent does not contain RNAs or DNase. It contains protein. Finally, in table 3, please take a look at that when you have a chance. It's an important table. Table 3. Prusiner shows that in order to inactivate the scrapey agent with UV irradiation, you need to irradiate it with 42,000 joules per meter squared. And in that table, he shows that viruses are inactivated with a much, much lower dose of UV irradiation. So, for instance, bacteriophage can be as low as 4 joules per meter squared irradiation. And the toughest critter that they looked at was the potato spindle tuber viroid, which is inactivated or killed at 5,000 joules per meter squared. So in doing so, Prusiner proves that the prion disease, the scrapie agent, is not a slow-acting virus, but rather a protein that he named prion. Okay, everyone, that's it for today. Please stay safe, and talk to you tomorrow.